Different types of muscle employ different mechanisms for excitation-contraction coupling. My aim here is to explain each mechanism and compare and contrast their properties. In case you're not already familiar with the different types of muscle, smooth muscle is found in the walls of hollow organs, such as the stomach, intestines, bladder, airways, and blood vessels, and it's responsible for constricting the organ to control the movement of food, fluids, or air through them. Cardiac muscle is found only in the heart, where it is responsible for contracting the heart's chambers to eject blood into the circulation. Skeletal muscle is attached to bones throughout the body and contracts to bring about locomotion. For example, when we walk, raise our arm, or talk. As you can see here, the three types of muscle cell look very different. Skeletal muscle cells have a distinct striated appearance due to the ordered arrangement of the contractile proteins inside the cell. Skeletal muscle cells are also known as fibers, as they are quite large with a long cylindrical shape. Each cell or fiber spans the length of the muscle bundle and can be tens of centimeters long with a diameter of around 100 microns. Another feature of these cells is that they contain multiple nuclei. Cardiac muscle cells also appear striated due to the pattern of contractile proteins. They are not as large as skeletal muscle cells, but compared to other mammalian cells, they are quite large, at around 100 microns long and 25 microns wide, and they look distinctly rectangular. They can contain either one or two nuclei, and the ends of the cell contain a structure known as an intercalated disc, which holds neighbouring cells together as the muscle contracts. Smooth muscle cells lack the striations of the other muscle cells because the contractile proteins are less well organised, so they have a relatively smooth appearance. They are a lot smaller than the other muscle cells. Their dimensions vary in different organs, but they are all thin and spindle-shaped with diameters of a few microns and lengths ranging from around 30 to 200 microns. Smooth muscle cells all have a single centrally located nucleus. There are some commonalities in the mechanisms employed by the three muscle types for excitation-contraction coupling. In each case, contraction depends on the activation of voltage-gated calcium channels in the plasma membrane. These ion channels are activated by signals that cause depolarization of the membrane, although these signals differ among the cells. Calcium channel activation leads to a rise in the intracellular calcium ion concentration, and that, in turn, causes the muscle to contract. However, the steps between calcium channels being activated and the rise in intracellular calcium ion concentration is different in each of the three muscle cell types. Excitation-contraction coupling is arguably least sophisticated in smooth muscles, so I will start by explaining the mechanism in these cells. The calcium channel that mediates excitation-contraction coupling in smooth muscle is Cav 1.2. It opens when the membrane depolarizes, and this allows calcium to enter the cell where it comes into contact with and binds to calmodulin. The activated calmodulin forms a complex with myosin light chain kinase, which in turn phosphorylates myosin to evoke cross-bridge formation between myosin and actin to generate contraction. The contractile proteins then pull on the cell in all directions, causing the cell to round up. Cardiac cells have evolved a structure to facilitate the spread of calcium around the cell and deep into the centre of the cell. That structure is a network of T-tubules, which are invaginations of the plasma membrane that extend deep into the cell. Immediately under the T-tubules is another network, the sarcoplasmic reticulum, or SR, which acts as an intracellular store of calcium. These networks surround individual bundles of contractile proteins, or myofilaments. The closeness of the plasma membrane, the SR, and the myofilaments means that they can act as a unit. As in smooth muscle, the calcium channel mediating excitation-contraction coupling in cardiac cells is Cav1.2. But this time they work in coordination with calcium channels in the SR membrane, which control the flux of calcium out of the SR store. The calcium channels in the SR are known as ryanodine receptors, as they were first identified from the ability of ryanodine to activate them. They are encoded by the RYR2 gene. The Cav1.2 channels are strategically localized in the plasma membrane directly opposite the SR ryanodine receptors, which are only a few nanometers away. Excitation starts with an action potential that travels along the cell membrane and into the T tubules, where it causes calcium channels to open and calcium enters, as in smooth muscle. But this time, the calcium binds to the immediately adjacent ryanodine receptors, which open and allow calcium to flood out of the store immediately next to the myofilaments. Because this happens simultaneously throughout the T-tubular network, a coordinated contraction results. Skeletal muscle cells also contain a T-tubular network, and it works in coordination with the sarcoplasmic reticulum to generate coordinated calcium signals throughout the cell. Excitation-contraction coupling in these cells uses Cav1.1 channels to coordinate calcium efflux from the SR through ryanodine receptors. This time they are type 1 ryanodine receptors. The mechanism of coupling between the calcium channel and ryanodine receptor in skeletal muscle is, however, quite different. First of all, you should be aware that the Cav1.1 channel doesn't actually pass much current, so it is not a very effective calcium channel. That is the reason the skeletal muscle action potential remains brief despite calcium channels being activated. The skeletal muscle calcium channel is often referred to as a dihydropyridine receptor because it was first identified due to dihydropyridine binding, at a time when no one had been able to identify a calcium current in the cells. So there was a lot of controversy over what the binding actually represented. But now the problem has been resolved. We know that the channel is present, but the skeletal muscle calcium channel acts simply as a voltage sensor and not as a means of mediating calcium influx. In fact, a cytoplasmic part of the Cav1.1 channel, located in the linker between the second and third transmembrane domains, directly interacts with a cytoplasmic region of the ryanodine receptor located in the sarcoplasmic reticulum membrane. So the mechanism of excitation-contraction coupling in skeletal muscle is as follows. An action potential travels along the plasma membrane and down the T tubules, where it activates Cav1.1 channels. These channels undergo a conformational change, but instead of causing channel opening and calcium flux, it results in the cytoplasmic region attached to the ryanodine receptor pulling on the ryanodine receptor, much like a coiled spring. This pulls the channel in the ryanodine receptor open, causing calcium to flood out and activate the nearby myofilaments to generate a coordinated contraction.